Okay, so I just mentioned that that the immunohistochemistry in the microscope. Remember seeing the pictures of cells and they're like red and green stained and stuff. So those are fluorophores or fluorescing markers and stuff. Well, you could add, you know, some kind of a chemical fluorescent thing that would glow, something that would glow and allow you to see a piece of DNA. And so actually on the PCR reactions, this type of PCR reaction is called endpoint PCR. And you make a piece of DNA of a specific size, you run that on a gel, and this glows because you're adding a chemical called ethidium bromide. Ethidium bromide interchelates into DNA and it glows orange with UV light. So you can see it that way, right? So the people that were trying to think of different ways to do this other than PCR were thinking, well, you know, if we use like an ethidium bromide type of molecule, you could look at the accumulation of this new DNA and it would be glowing at a certain wavelength, just like ethidium bromide. And we could add an electronic detector, a fluorescence detector with a filter so it recognizes that wavelength of light, and then it just counts it, quantitates it, gives you a printout of that. That's kind of cool. So then you have like a machine that's measuring this whole reaction. You don't need to run the gels, and then it turns out that the gels are not very quantitative anyway. When they first started to look at the kinetics of PCR, what they saw was that if you look at the amount of DNA made per cycle, this is the cycle number. And what do I mean by the cycle? It's the number of times that the DNA was heated to boiling, and then the polymerase was able to make one new copy. So then that's one cycle. So you go from 95 degrees where it's cooking and you cool it down. You let the primers hybridize, and then also the polymerase can start to copy at that temperature. And so that's one cycle. So you look at the cycle number. Usually you don't see very much for the first couple of cycles. And that's because, you know, it takes more than just a few molecules to be able to see something, right? The level of detection that we're at, you need to run, you know, quite a few cycles. You know, we're not going to see 2 molecules going to 4 molecules, going to 8 molecules. It's like 8 molecules in a drop. The molecule is pretty small, and there's not a lot there to see. But it's when you get into the millions where you start to really see fluorescence. So what ends up happening is that you get this sort of curve. So, and then this number is somewhere between, it's in the 20s to 30s usually, number of cycles. That might sound like a lot of cycles, but 2 to the, you know, we're saying that 2 to the 4th power is, 2 to the 2nd is 4, 2 to the 3rd is 8, 2 to the 3rd is 16. You know, so I mean, it starts out slow, but it's when you get up into the thousands where, you know, doubling that number and doubling, it gets large very quickly, right? Logarithmic growth. So it takes 20 to 30 cycles to really start to pick up signal, and actually there's a threshold value that the machine can start to see the signal. And that threshold value for the cycle number, they call that CET. So if you compare the curves from a couple of different samples, like if this is the untreated sample, right? So RNA that you made, and you make it into cDNA, and then you got this happening. That's untreated. You know, it could be that the treated sample is going to look like this. Looks very similar. Right? treated with TGF-beta. So it has a similar curve to it, but the cycle number that it actually hit the CT value at is going to be different. So let's say the CT value was 26 as an example. 26 for the untreated sample, but 22. Cycle 22 for the treated sample. The whole curve shifted over. So that means that there's four cycles difference between the two samples. So it's 2 to the 4th power. So it's 2 times 2 times 2 times 2, which is 16. So that means that there's a 16-fold difference in the amount of the gene there. You get that? So this is a lot of stuff, and this is view this as your first exposure to it. And I'm pleased to provide you with a lab and some new equipment and stuff that you can really get the feeling for what the modern application is for PCR. But as far as understanding all the nuances of it, you're going to get this material over and over again in other courses, and so there's no way that I can go over all that in a short amount of time. But I think you got the flavor of it, right? And so, there's some specific details, such as, how do they detect this fluorescence? And so they talk about this in various videos, and we'll watch these in the lab. Basically, these are DNA strands, and if we have the complement over here and here, the primer, and then the new DNA synthesis is occurring. You can either add a dye that lights up that DNA, or it'd be even cooler if you could play around with different fluorophores in a more advanced chemical reaction and detect it that way, kind of indirectly. Because if you just look at the amount of DNA, you have DNA template there to begin with, and you have DNA primers, and then sometimes you get some spurious amplification events like primer dimers occur, and so there are other size fragments that are not the thing that you're interested in. So if you just use dyes on this, there's some chance that you'll get a wrong result. And the dye that they use right now is called Cyber Green, and that is used in real-time PCR machines. It replaces ethidium bromide. It's a better way to look at the amount of DNA that's being made. So Cyber Green is just a dye that's going to sit in DNA, and it's going to light up because there's DNA there. But as I said, it's going to sit in any kind of DNA that's present. And so there was the primer, the original template DNA that you added, and then if there's any other fragments that start to form, it will light those up too. And if you're only detecting fluorescence, if you're only detecting the glow of light from these from this dye, then anything that glows, you won't be able to tell the right stuff from the wrong stuff. So anyway, so there's this clever company, local company called ABI, Applied Biosystems, and one of the fun things about studying this stuff is a lot of this stuff was invented in the Bay Area. You notice how, I say the guy that won the Nobel Prize for PCR, Kary Mullis, is a local Bay Area guy. The company he worked at, Chiron, was bought out. Started out as Cetus actually, and Cetus was bought out by Chiron, and Chiron was bought out by Novartis. But they're still located over in Emeryville, in the East Bay. So a lot of this stuff was invented right here. And it's hugely important stuff. Anyway, so the Cyber Green is one way to go, but there's a different way to run the reaction, and this is kind of fancy. So, so if you create a reporter gene, a reporter probe thing, so this will also hybridize in this DNA. So remember, we're amplifying up a discrete piece of DNA here. And we've got the primers, we're about to amplify this up, but we've created another primer that just sits in the middle somewhere. Okay, it sits in the middle, and we put two different fluorophores on it. And you'll see this in the videos. So, I mean, it's a lot more elegant than me drawing it. But if the reporter has two different fluorophores that are glowing on it, or have the potential to glow, one of the fluorophores is the thing that you want to glow, and the other one is actually going to absorb light from the first one. So that one's going to be called the quencher. So this quencher, you got this reporter primer thing, and it's going to sit in the middle of the thing that you want to make, and it has a fluorophore on one end and a quencher on the other. So when those two molecules are next to each other, this thing, the glowing from this, you never see that because it's absorbed by the quencher, because they're so close to each other on the molecular scale. They're like sitting right next to each other. So this one absorbs any light coming from the first one. But as soon as those things are clipped and they float apart, then you're going to see the glowing from this first one here. And so that's actually how this machine that we have works. And they call that TACMAN. So the TACMAN chemistry, and it's a company name, like Pipette Man or Walkman, I guess. Sony Walkman started out, and then people started adding they want to name their invention or whatever. So TACMAN is a invention for quantitating the PCR using a reporter. So this is the reporter. A reporter that contains a fluorophore and a quencher, and it's only when those two things are clipped apart do you see a signal. And it just so happens that the DNA polymerase, as it goes along, and it sees this little primer in the middle, it just chews that thing up. It chews it up because the DNA polymerase has an exonuclease activity. I mean, a lot of these enzymes that are working on DNA and RNA, they're multi-subunited, pretty complex little machines. And so if there's something in the way, they just nibble that stuff out of the way. It's like, that just goes away pretty quickly. So it will clip this thing down to little pieces very quickly. And so when that happens, those two molecules separate. The fluorophore and the quencher separate. So suddenly the fluorophore is just floating free in solution. Well, it's giving off light. Well, we've got this machine that's very sensitive, and it's calculating how much light does it see. It draws you a line. It's like I see more and more light over time. The more cycles of the PCR that you run, the more light you're seeing. And it just gives you a plot. That's what the light looks like. So, does that make sense? So you'll see it, illustrated on the videos, and I think they do a really nice job with this. I don't think I need to belabor that. Okay, so we're just about done. The one thing that we haven't really talked about is the cDNA reaction. It's like, why do we have to get involved with all that? I could have talked about that first, but I didn't want you to be bored to tears. So right now we have RNA in a tube, right? You guys prepared RNA. And if you missed the lab last week, you need to jump in with some other folks that have prepared some RNA and be part of that. Okay, so we have RNA, and we have two samples. You have the untreated and the treated one. You have the one that was just from fibroblasts, and then you have the one that was from fibroblasts with TGF-beta added for a couple of days. The TGF-beta was around, so it stimulated collagen production. So you made the RNA. And you have these two samples. We can't use PCR and RNA. Oops. So it's just, it's just the way it happens, right? RNA is a pretty unstable molecule, by the way. Right? And remember me saying that like the tips of your fingers have RNase activity? Remember the word ACE is an enzyme, so RNA, ACE. So it's an enzyme that digests up RNA. It's going to break it down. So that's pretty it's kind of a bad thing. RNA hydrolyzes really easily. The nucleotides can be hydrolyzed and the whole thing just comes apart. So RNA is a photocopy of the original, but it's not meant to be a very valuable photocopy. I can print off a bunch of these things on the photocopier, but like this particular copy doesn't really mean much to me. And so, RNA is kind of flimsy then. I mean, it's not used as a template by a DNA polymerase. If the template was RNA, then actually it's used by ribosomes as a template to make proteins. That's what RNA is actually used for, right? So it's not used as a, ordinarily as a template to make more RNA molecules. So in order to quantitate the RNA, we have to make DNA of some type. It's more stable and we're able to replicate the DNA. Now, could we just look at the RNA? Yes. Like when I was in grad school, we didn't use PCR. We would isolate RNA and then we would analyze the RNA. We could run RNA out on gels, and then to see it, we would have these things we call probes that would be radioactive. So we'd have a radioactive complement to the RNA, and then you run it on a gel and you can see the band glowing. But everything was so radioactive, we always had Geiger counters at our desks, and we would, I think I talked about this before. I mean, we would just notice that there were radioactive spots on our desk, and sometimes radioactive spots on the floor, and sometimes a pen would become radioactive because somebody touched it that was working with some isotopes or whatever. So radioactivity was all over the place, which is pretty dangerous stuff, right? So we don't want to work with radioactivity, and we want to be able to detect how much RNA was made. So we can use PCR if we make a copy of the RNA into DNA. Once you've got a piece of DNA, you can amplify it with PCR. So that's great, the good news. But you have to still go from RNA to DNA. Well, you remember from the central dogma of molecular biology, what is the central dogma again? Tam, central dogma. DNA is transcribed into RNA and then what? Translated to protein. Yep. See the way she said that? That was perfect, right? That this process here is called transcription, so transcribe. And that this process here, RNA being translated into protein. So if you confuse these two terms or use different terms, you won't get any credit on an exam, because those are the accepted ways to refer to those processes, have a very specific definition, which is one of the sort of buggy things about biology generally, right? Like there's a whole bunch of specific terms, they have specific meaning. If you have the word a little bit off, or the value a little bit off, the equation off a little bit, whatever, then that's not worth anything. You have to have it be exactly right. So we're saying that we want to go from RNA and create DNA. Hey, wait a second. That doesn't fit in with the central dogma of molecular biology. DNA goes to RNA. RNA doesn't go to DNA. So how do we get that? How do we get it to go backwards? Well, it turned out in nature that there's, just like every time there's a rule, there's an exception to the rule. You ever notice that? Sometimes people say that the exception proves the rule. Once there's a couple of exceptions, then you know that the rule really is a rule. It's just that there's always going to be some more minor examples of things going the other way. So it turns out in nature, it can go the other way. It's an exception to the rule. It's not very common, but it happens. So when does that happen? It happens in viruses. There are certain viruses that have an RNA genome, and at a certain point they copy that into DNA. And it's because they want the DNA to integrate into the host genome. So you could get sick with a virus, and the virus might have an RNA genome, and it will make DNA, and then the DNA could integrate into your cellular DNA. kind of disturbing, right? And then once it does that, I mean, it's hard for the body to detect that there's been some new instruction coming from a virus inserted. How often does this happen? Well, it turns out that like our genomes are littered with viral sequence, and so we're all messed up, but that's okay because it maybe is driving evolution to begin with, right? So all of this extra copies of DNA and all that stuff, weird things going on in the genome. Viruses are messing with our genomes all the time. So this virus, these are called retroviruses. And I'll give you an example of a retrovirus, HIV. Okay, so a certain category of viruses known as retroviruses can take an RNA genome and make DNA. And they do that with the idea of possibly integrating into the cellular DNA. And so those retroviruses, an example is HIV, which we all know, right? And it's a pretty scary virus, etc. And there's a couple of others too. But anyway, these viruses, they code for proteins. You know, it's once something has a genome, that means it has an instruction set. And what does the instruction set do? The instruction set typically codes for some proteins. So it has some genes, and it's going to make a couple of, you know, protein machines, a couple of enzymes or whatever, right? That's going to allow its genome to replicate. Isn't this weird stuff? I mean, this gets pretty weird. But this is the stuff that you're studying, and this is so mainstream at this point, it's amazing. But people have been studying this for several decades, so the contributions coming in from different types of science, different people, virologists looking at viruses and saying, oh, look, you know, there's sometimes RNA can go to DNA. And then some of the molecular genetics people are like, well, we're interested in that because we're tired of working with RNA. It breaks down really easily. Can you give us this enzyme, this retrovirus enzyme that takes DNA to RNA? And yeah, they purified it out. They purified out an enzyme. And so the enzyme is able to take that RNA that you have in the tube and make some DNA out of it. And it's called reverse transcriptase. Right? That's a pretty rational name for it. Pick that apart. Please remember that. Reverse, because you're going from DNA to RNA. You're going in the reverse direction. So it's a reverse transcriptase. This process of DNA to RNA is transcription, right? So it's a reverse transcriptase. ACE is an enzyme, right? Reverse transcriptase. So you should be able to tell me that enzyme. So that's the enzyme you use in class to take your DNA, your RNA sample, and make DNA. And we call this, by the way, cDNA. Well, it's a something special that we do in labs. So it's called cDNA. Sometimes copy DNA. Like it's a copy of the RNA, but it's a DNA copy. So we call it cDNA. So before we can get going with this whole PCR thing, you're going to need to take your RNA samples that you have in the tubes, and you're going to add a couple of reagents, and we're going to incubate this, and it's going to make cDNA. And then during that incubation period, we're going to be watching some videos about polymerase chain reaction ad nauseum until you sort of feel like, okay, I get it. And then we're going to pop it into the quantitative PCR machine. So Applied Biosystems, qPCR, and it's like a computer-looking thing, and you've got this plate of different wells. It's like a bunch of plastic tubes connected to each other, and we're going to add the PCR mix to the sample, to your cDNA sample, and we're going to run it in the machine, and we're going to get those curves that are showing cycle number and amount of product that's being made. And you're going to see these lines, and they'll be close to each other, but you know, they're off a little bit. That could show several fold increases in the RNA. So it gives us a quantitation of the RNA. So that's it. That's the crux of the whole thing. What do you think? Some questions for me about this? This is a big chunk of molecular biology. So, you know, if you can really appreciate what's going on in this lab, it's going to serve you well in all your other classes. And it's not a minor thing because it's the main diagnostic tool for analyzing DNA and gene expression. So, you know, we're in the age of molecular medicine. You go to the doctor, they're going to start analyzing your DNA more and more and your gene expression levels, and they'll be doing exactly what you're doing in the lab, basically. So, I mean, this stuff is all over the place. And it's also used for all of the genetic engineering stuff. And I don't know, are you guys going to vote on the genetic, you know, there's a proposition, what's it, Prop 37? Did you hear about that? You know, should we label the food that we eat? You know, is it genetically modified or not? I don't know if I'm supposed to, you know, try to influence you. I mean, personally, I'm actually worried on the genetic modification thing. I think we're playing with things that we don't fully appreciate. I mean, the genome, this stuff is not a joke. You know, DNA, RNA, this is the code of life. Okay, just to put it in perspective, it's the code of life, and we're now able to manipulate the code of life, and we've done that for a bunch of different things that we depend on. We have industrial processes, like if you use paper, this paper is all processed using genetically modified enzymes, etc. So, I mean, our whole industrial production is shifted towards genetic modification, and then the food that we eat is largely genetically modified. In the United States, like the corn that's genetically modified is 90-something%. The soybean that we eat is over 90% genetically modified. I mean, most of the, you know, the real staples, heavy staples, have been genetically modified. So my take on it is that, you know, some genetic modifications, if they're done well, if they're done cleverly, it could be a tremendous help. You know, and if you're living in a world where superstorms wipe out New York City and things like that, tornadoes, you know, ravage the Midwest corn belt, and there's a, you know, drought there right now too. You know, that kind of stuff, you're going to need to be, you know, have some new tools to be able to get around some of these problems. On the other hand, just because you can engineer something doesn't mean it's engineered well. You can engineer something kind of foolishly or intelligently. And so there's always going to be a bunch of people that do something stupid, and then it's going to create a problem. So then you have to put some regulations in place to limit the stupid stuff that was done, and be smart about realizing when something is damaging your environment or whatever. And then the other things that are good, those things can be very helpful and help feed the fact that you've got, you know, over 10 billion people on the planet or whatever, right? So you're going to need ways to be able to handle that. So that's kind of my take on it. So I think, I don't know, in the short term, I think I'm probably inclined to say we should know what's genetically modified, just because there's a lot of stupid things that are initially done. But in the end, I mean, in the end, we need some genetic modification of industrial processes and food production. So that's kind of where I'm at. Any questions on this stuff? So there's a lot of stuff here. So we start the reactions in the lab. Start with your RNA tubes. We make the cDNA, then we watch a bunch of videos. Once the cDNA is done, we have to go down to the nanodrop machine. Remember this? So you get a measurement again. Did you make any cDNA? And once we verify that there's cDNA, we mix it with the reagents that we need for PCR. We pop it into the PCR machine. That takes another 30, 40 minutes. And then once we get that, pop that into the computer, and it gives us these curves. It shows how much expression we have. From our perspective in the lab, you guys are actually doing an experiment that we really desperately want to do. So, you know, like in some labs that you'll have at this university, they want to demonstrate something. We're demonstrating something where you're actually doing real research, because we want to visualize how much RNA is being made for our collagen. We added TGF-beta. What does the induction look like on the collagen level? So you're going to help answer that for us. So I'm interested, keenly interested in some of your results to see if you can see that increase in the gene level for collagen. Okay, go to the lab.